Let's talk about the announcements. Meet in the lobby at 1. They also asked that people wear closed-toe shoes. They don't have to be steel-toed, but they don't want people in sandals or open-toed shoes. I've adjusted the due date for homework 11 compared to what's published on the course schedule. You know, the course schedule indicated that that assignment would be due on Wednesday, but I've updated it to be due before class on Friday. So homework 11 is available now. For the two most calculationally intensive problems, I've put the answers so that you'll know whether you're on the right track. There's one problem related to aeration for secondary treatment, another one related to trickling filters, which we're talking about today. So those have the answers on them. The others do not have the answers. We're going to have our exam in class on Friday, and I'm providing the formula sheet. In fact, it's available now on Blackboard if you want to get a look at what's on there. So it's at the bottom of our class page. It's called Exam 2 Formula Sheet. So it's got some equations that I hope you'll find familiar. It's got kinetic reactor type formulas. It's got mixing power equation formulas, settling velocity, BOD removal. Not everything on here we've talked about. You know, for example, I don't think we talked about dilution when it comes to BOD testing, but it does have the aeration basin BOD removal equations that we've just been working through. And then down here at the bottom, these are the trickling filter equations that we'll start talking about today. It's got some sludge equations. And so I'll provide this formula sheet. You don't need to bring any notes, and in fact you won't be allowed to use notes on the exam. So it'll just be bring a calculator, bring a pencil, and I'll provide the rest. The coverage is going to be classes 17 through 29, just basically the material since our last midterm exam, plus the water treatment plant tour that will be going on Wednesday. Any questions related to the announcements? I emailed the solution to the in-class exercise related to sludge production. Here's a printed copy of it on the back side of today's in-class exercise, just so you'll have it on paper. And it's the solution to in-class exercise 28 we use as a starting point for today's in-class exercise as well. Everybody get a copy? All right. So today we're going to be talking about secondary treatment with attached growth. And remember, secondary treatment is primarily focusing on the removal of soluble BOD. It's the BOD that won't settle. And so these are just different ways to get that BOD reduced. And if we're doing it in an aeration basin, we have to have some way of providing oxygen to the wastewater. And so mechanical mixing would be to have a motor and impellers which are mixing the mixed liquor and bringing it to the surface with enough energy that it's kind of splashing around and entraining air. And mechanical mixing like this is less common than using air diffusers. And so what we saw at the Huntington wastewater treatment plant was air diffusers where it's a membrane that has very small diameter pores, and they're using big engines or motors to blow air through the diffusers. And this is what it would look like in an aeration basin when it's drained, just a series of diffusers. And to try and prevent anaerobic conditions, then it's important to keep the oxygen between 0.5 to 2 milligrams per liter. Any less than that, and you risk there being portions in the aeration basin that don't have any oxygen, and if there's an anoxic zone, then that can be, it can produce really bad odors, and the rate of BOD removal slows down dramatically. A typical rule of thumb we've talked about before, that for every 1 meter of every 1 cubic meter of wastewater, about 8 cubic meters of air is provided. So if the water is spending a certain amount of time in the aeration basin, that would tell you, that would indirectly give you an idea of the volumetric flow rate of air into the aeration basin. So in the time that the average fluid particle spends in the aeration basin, there should be 8 times the volume of air that's provided. And to be more specific and scientific about it, this formula expresses the exact oxygen demand that's required. And so we provide more oxygen than actually the microorganisms are using, because some of it is going to escape. It'll get to the surface and it won't have diffused into the liquid. And so this formula tells you actually the microorganisms that are there, how much oxygen they are consuming. And we're providing a lot more than they consume, because not all of it goes into solution, but this formula tells us that it's related to the flow rate of the water that's being treated, and then the difference between the incoming BOD concentration and the outgoing BOD concentration. And so this difference, S naught minus S, you can think of it as how much they ate. So how much oxygen you need to provide is how much BOD they consumed, and we're expressing how much they ate in terms of oxygen demand. And so it's very easy for us to calculate the mass of oxygen required when we're expressing their food consumption in terms of the oxygen that it demands. And then also this F term in the denominator is the ratio of the BOD5 to the ultimate BOD. And the reason why we have to include that is because usually S naught and S are expressed in terms of the 5-day BOD demand, and it's the ultimate BOD that reflects how much oxygen is going to be consumed. And so it's just kind of a correction factor, because we're measuring oxygen demand in just a 5-day period, but it's the ultimate oxygen demand that impacts how much oxygen is needed. And then you can see that we're subtracting the amount of sludge production from the oxygen demand, because if they're generating sludge, that's cellular mass that reduces the oxygen requirements, because this difference in substrate would be assuming that it's 100% of the oxygen demand went into just cellular activity, and the mass of new cells that are produced reduces the oxygen that they are consuming. So here are the calculations that's on the front page of the in-class exercise handout that I've just given you. Let's say that this is an extension of the scenario posed in in-class exercise 28. We had a flow rate of 12,960 cubic meters per day. The incoming soluble BOD 5 is 84 grams per cubic meter. Remember that grams per cubic meter is the same as milligrams per liter. Just they're off by 1,000 in both the numerator and the denominator. So 84 grams per meter is what's coming in. The secondary treatment is knocking that down to 11.1 grams per cubic meter. And then we know that the sludge that's being produced in this aeration basin is 436 kilograms per day, and that's the dry mass of sludge solids that are being produced. So that's not the volume of sludge, that's how many sludge solid, the mass of sludge solids that's being produced. And if the ultimate BOD is 123 milligrams per liter, then we can use that to calculate F. So I'd like to pause for a second, give you a chance just to play around with this formula, see if we can determine what's the mass of oxygen that's required by the aeration basin, and that's going to be expressed in terms of kilograms per day. And then part B is a little bit more tricky. We'll do it together. It's just saying that if we assume that air is 23.2% oxygen, and we know the air density, then what would be the volume of air that's required, assuming 100% transfer of the air into the liquid? So just do part A and we'll do part B together. All right, so on the whiteboard I showed that F is the ratio of the 5-day BOD to the ultimate BOD. And so the waste that's coming in, it's common to just assess how much oxygen demand there would be in a bottle test in a 5-day period, 84 grams per cubic meter. But when the water is in the aeration basin, we're trying to use up all the BOD that's there, not just the 5-day BOD. So we actually have to provide more oxygen than the 5-day BOD would maybe indicate, because we want to break it down so completely that when the water is discharged to a stream, that it's not going to affect the aquatic life, and that's based on the ultimate BOD. So F, that correction factor, we first have to calculate, and so it should be 0.683. So to find the mass of oxygen that's required, then it's going to be the volumetric flow rate of the water that's going into the aeration basin, the difference between the inflow and the outflow, BOD concentration, and then a conversion factor here because I want to know the mass of oxygen in terms of kilograms per day, not just grams per day. So dividing by 1,000 effectively, or multiply by 10 to the minus 3. Divided by the correction factor F, and then subtracting out 1.42 times the sludge production. On the back side of the paper I gave you is that same solution that I emailed on Friday. It's just showing for these conditions, it's generating 436 kilograms per day of volatile suspended solids. So that volatile suspended solids, it's the increase in cell mass, and so if the solids are volatile, that means that they could be burned in an oven. It's representative of the increased microbes per day that are being generated by consuming BOD. So it reduces the amount of oxygen demand. So you can see 764 kilograms of oxygen per day. Now that's the mass. What if we tried to determine a volume from that? Just knowing that air is 23.2% oxygen and the density of air at sea level is 1.185 kilograms per cubic meter. So if the air is 1.185 kilograms per cubic meter, and we multiply that by 0.232, then that suggests that there's 0.275 kilograms of O2 per cubic meter of air. So we're getting 0.275 kilograms of oxygen for every cubic meter of air. And so how much air do we need? Well, if we need 764 kilograms of O2 per day, and divide that by 0.275 kilograms per cubic meter, then that means we need 2780 cubic meters of air required. So 2780 cubic meters of air required is much, much less than the 8-to-1 ratio I previously told you about. You know, the previous ratio of 8-to-1 was just that rule of thumb that, where was it? For each cubic meter of wastewater, about 8 cubic meters of air is required. So what was our volumetric flow rate in this example? The water is coming in at 12,960. So 12,960 times 8, 12,960 multiplied by 8. The rule of thumb would say we need about 104,000 cubic meters of air per day. This is saying only 2,700. The reason why is because most of the air that is injected at the bottom of the aeration basin floats to the top. There's very poor exchange or diffusion of the oxygen into solution. And the microorganisms are consuming dissolved oxygen. And what we've just calculated here, this is the volume of air that's required. So what that kind of tells us is if it's 2780 cubic meters and the rule of thumb was suggesting we needed about 104,000, it's about 2% of the air that is diffused at the bottom of the aeration basin is going to actually go into solution. And we can improve that, like the smaller the air bubble, if it's a very, very fine air bubble with a small diameter, then that increases the transfer efficiency. Any questions related to this example? All right, that's all we're going to talk about for suspended growth. Suspended growth means that the bugs are floating around in the aeration basin. They're suspended. And so they're floating freely, they're just consuming the BOD, consuming oxygen, and then being recirculated as the sludge settles in the secondary clarifier. So suspended growth is what we've been talking about so far. Fixed growth is where instead of floating around in a tank, the microorganisms are actually living on a surface as a film. And so that thin film, which is fixed on either rock or plastic or sheets, water is sprayed over that film. And so the water is moving, but the microorganisms are stationary, and they are taking the food from the solution that's being sprayed on them. And then we actually don't have to pump air, or if we do, the ventilation requirements are much easier than they would be if we were having to dissolve the oxygen into water. So we're going to talk all about fixed growth for the remainder of today's class time. One type of fixed growth is a rotating biological contactor. So this is a picture of an RBC, where basically these are big sheets of plastic, which many of them are stacked together, and then there's, you can't see it, but this is like a long rectangular pool of water. So these RBCs are being rotated. Part of the time they're dipped down into the water, part of the time they're above the water and exposed to air. And so the microorganisms are getting, like, on the round trip cycle of being rotated. They're getting exposed to air, they're getting exposed to substrate when they're dipped down into the water, and what they'll do is that a slime layer will grow on this, and eventually it will slough off. When it becomes too thick and too heavy, then it'll fall into the water, and that's good because that's what we want. We want it to turn the dissolved BOD into something that will settle, because dissolved BOD won't settle. It's in solution, whereas the cellular material, like the slime that sloughs off of an RBC, it can be settled in a secondary clarifier. So just to give you a schematic view of what's happening, the water that comes off of the top of a primary clarifier goes into this series of pools where the RBCs are exposed to the water, they're consuming the BOD, they're exposed to oxygen, and there's very little electricity requirements. Like, it doesn't use much electricity to rotate these. You're not having to pump the water, you're not having to recirculate sludge, you're not having to provide aeration necessarily. So it's a very economical means of reducing the BOD. It doesn't have a huge amount of removal efficiency. Like, it's not as effective, but for the BOD that is removed, you're doing it without much of a cost because the aeration and pumping is so much less. Another means of fixed growth is a trickling filter, and it's the same idea here. That what you want to do is provide a location for slime to grow. and you're spraying wastewater on top of like rocks, and naturally the slime is going to grow on the rocks. Any place there's food, something is going to grow to use that food. Whether it's like, you put something in the fridge, that slows down the growth of bacteria, but eventually they're going to find the food, and something that's able to grow in low temperatures is going to spoil your food. So anytime there's like a substrate that can be utilized, some form of life is going to arise to consume that substrate. So what we've done is we've just provided a location that's favorable for microorganisms to break down BOD, and because it's a porous media, like it's a bunch of rocks stuck on each other, the air can naturally circulate through there. Maybe we'll put some fans in it to like enhance the exchange of air if needed, but it's a very low-cost way of removing BOD, because in some cases you can even use the gravity flow to cause the rotation and the spraying of the water onto the trickling filter. So just to give you an idea of where this sits, it would just take the place of the aeration basin. So we still have, a bar rack and screening. We still have a grit chamber. There's still primary settling. But then after primary settling, instead of sending the water to an aeration basin, you'd instead send it to a trickling filter. Now there is recirculation in a trickling filter, but it's recirculation of the supernatant, the stuff off of the top of the secondary clarifier. It's not recirculation of the sludge. And so that's a big difference between aeration basins and trickling filters. Is in aeration basins, you're recirculating what comes off the bottom of the secondary clarifier. But in a trickling filter, you're recirculating what comes off the top of the trickling filter. And the main reason for the recirculation is just to increase the contact time, because the water's going to drip down through this very quickly. And you may want the water to spend like an hour in the trickling filter. And so if it just drips down and it spends 5 minutes in the process of dripping down, and you want an hour of contact, that means you have to recirculate the water 20 times to get that average 1 hour residency time inside the trickling filter. So the flow rate through the trickling filter includes the flow that's coming in from the primary clarifier and then also the recirculation flow. So secondary treatment here is just providing a location for the microorganisms to do their thing. So here's a look at spraying over a circular trickling filter. Very commonly these are circular in shape. If you look down from above, cylindrical would be, I guess, the shape of it if you consider the fact that it's got depth. So water is just spraying over these rocks. They look like ordinary rocks, but then when the layer of microorganisms get thick enough, then it doesn't have the strength to stay attached to the rock. And so eventually just a layer of slime goes to the bottom, and then that can be settled in the secondary clarifier. Some of them can be really big as well. Traditionally, stone would be used as the porous media. More recently, they found that plastic is a lot lighter, and you can increase the specific surface area by using plastic. Specific surface area is in a cubic meter of volume of the trickling filter. How much surface area is there for the microorganisms to be growing on? And you don't want to make it so that the pores are too small, because then that would clog easily. And so there's kind of a happy medium between the plastic media size to allow the slime layer to get to the bottom. In the case of rock filters, they may be as much as 3 meters or 10 feet tall. You're able to go quite a bit taller if you use plastic media because it's less dense. So up to 12 meters deep potentially if you're using a synthetic media. And this is just a look at what it would look like. Cylindrical, hollow plastic tubes that are cut pretty short and have themselves holes in them. So the water would just seep over there, and you're kind of like watering plants in a sense, except for that what you're trying to do is get some bacteria to grow on that plastic media. Here's another possibility, is that you just drop in kind of more of a solid mesh frame, into the trickling filter. Some installations have had hanging sheets of plastic that they spray the water at the top and they inject air at the bottom. And as long as the air can circulate around inside of the filter, then the bacteria are happy because they have the oxygen that they need, they have the water that keeps them wet, and they have the BOD in the water. So the method of action here is just microbial growth accumulates on the media surface, the wastewater is wetting it, the microbes consume the waste that's in the water, and calling it a filter is kind of a misnomer. Because remember, when we talked about sand filters, what was the mechanism of action in a sand filter for drinking water treatment? Do you remember that from the exam? I asked you the mechanism of action. What was it? Good. Yeah, impaction or collision and then adhesion. So it's not actually screening. So that was just a first example of how, like, filter isn't what we normally think of. Normally when we think of filter, we think of screening. So in sand filters, it's not screening. And here, it's also not screening, not by a long shot. So calling it attached growth is maybe more accurate than calling it a filter because no screening occurs. The pores are much too big. What we're having is just some scum accumulate, and then eventually the scum layer gets too thick, sloughs off, and it can settle in the secondary clarifier. So we're turning dissolved BOD into something that will physically settle. And this slime, once it sloughs off, it will physically settle. The limitations of a trickling filter is that if the organic load is too high, meaning that if you have a really high strength waste, then it's possible that the slime growth would happen so quickly that it could clog the pores. And so you have to, select the size of the media after knowing the characteristics of the waste, including the temperature that's likely to occur. There are the possibility that if you don't have sufficient ventilation, that oxygen could be depleted in the trickling filter. And so if that's the case, then they'd ventilate it by turning on fans. And this is especially vulnerable to cold weather because air doesn't hold temperature as quickly, or as long as water. And so like an aeration basin will maintain, even in really freezing cold temperatures, it'll maintain roughly the same temperature as the water that came into the treatment plant, like mid-50s, 40s or so. But when you have a really thin layer of water, it loses its heat more quickly. And that's what we have here in trickling filters, is very thin layers of water that are more susceptible to cold weather. So the things that designers consider when they're sizing attached growth media is whether they're going to use a rock media or synthetic like plastic. And so plastics offer the increased surface area per meter of volume compared to rocks. It's also lighter, and you can get better air circulation through the synthetic media. Trying to decide how big the trickling filter should be and its shape is one of the factors that they'll think about. The hydraulic loading rate is trying to make sure that you keep the slime from drying out. Applying enough water so that you don't have it prematurely sloughing before it's a thick enough layer. So there's a minimum wetting rate that can be difficult to maintain at night if you don't have a flow equalization basin. Flow equalization basins can also give you some control over the organic loading to keep that more constant during the day. And then how much of the waste gets recirculated to increase the contact time is another important factor. So recirculation is just the idea that of the water that comes off the bottom of the trickling filter, some of it is going to go back into the top of the trickling filter and some of it will be discharged. So how much recirculation you do is a means of improving the contact time and increasing the BOD removal efficiency. And it also, during low flow rate periods, you can increase the recirculation ratio and then prevent the filter media from drying out. The other nice thing about recirculation is that the water that comes off the bottom of the trickling filter usually has a much higher dissolved oxygen content than the water that's coming in from the primary clarifier. So the recirculated water increases the dissolved oxygen of the mixture that gets into the top of the trickling filter. And it can make it easier to spray and make sure that you've got adequate coverage over the entire surface area of the filter. So there's a parameter that will calculate the recirculation ratio, which is the flow rate of the recirculated stream relative to the overall flow rate going through the treatment plant Q. And it's common to use trickling filters in sequence to have, for example, well, you could have them in parallel and also in series together. What this is showing is that they're having the water coming off of the primary clarifier, and then the stream is split two ways. Half of the flow would go through one of the first plastic media towers, half of the flow is going to the other, and then it goes through a clarifying process where whatever sloughing occurs in the primary first stage trickling filter will settle to the bottom, and that's where the waste sludge comes from. But then the water that comes off of the intermediate clarifier, some of it's recirculated, but then it's sent through a second trickling filter to further polish and bring down the BOD even more. So these are the design equations that we use for trickling filters, and they're just strictly empirical. That means that they were developed based on observation and not based on like first principles of physics and biology. It's just they observed a lot of trickling filters and kind of calibrated the performance over time to a couple of coefficients that are built into the equation here. So let's use these equations to consider this trickling filter example. Let's say that we've got a two-stage trickling filter. So water comes in to one, part of it's recirculated, part of it goes to the second filter, through which there's recirculation, and then the water ultimately goes out. We know the diameter of each of these filters and their height. So from that, we'll be able to calculate the volume of the filter, which is an important parameter here in calculating the removal efficiency. We know the flow of the wastewater and the flow of the recirculation, which that defines R for us. And then we've got the concentration of the BOD coming in, and we know the temperature. And by the way, these equations are calibrated for 20 degrees Celsius. And so if you have a temperature other than 20 degrees, then you have to apply a correction factor before calculating the effluent concentration from the trickling filter. All right, so the first thing that we do is we would calculate the area based on the circular profile of this filter. We know the diameter of it, and use the diameter that's given to find the cross-sectional area, and then the filter is itself cylindrical, so we'd multiply the area by the height to find the volume. Okay, so the area is pi times the diameter of 17 meters squared divided by 4. That means the area here is 226.98 meters squared. And then the volume would be 226.98 meters squared times the height, 2.5 meters. And so the volume of the filter, 567.45 cubic meters. So we're going to use that here in E1 and E2. We assume that they both have the same size, just for simplicity. The recirculation ratio, R, is Q sub R divided by Q. So the recirculation flow rate is 0.017361 cubic meters per second, and the flow rate is 0.006944 cubic meters per second. So the recirculation ratio is 2.5. That means that there's 2.5 times as much water going up to the top of the filter as is coming in and coming out because of recirculation. So that's the recirculation ratio. The recirculation factor, F, is 1 plus R divided by 1 plus 0.1 r, and all of that in the denominator is squared. Okay, so it's 1 plus 2.5 divided by 1 plus 0.1 times 2.5, and then all of that is squared. So our recirculation factor is 2.24. Okay, so if you look at this formula E1, we know the flow rate of the water that's coming in. That's this 0.006944. The concentration of BOD coming in is 160 milligrams per liter. The volume of the reactor is defined by its diameter and height, and the recirculation factor F is known as well. So let me bring open, by the way, these calculations just so we get it in the video. Okay. So when we put the flow rate, the concentration, the volume of the reactor, and the recirculation factor, what this E is telling us is it's the efficiency of removal. So this trickling filter is removing 89.14%. In other words, as a fraction, it's removing 0.8914 of whatever was coming in. But that is the efficiency, that's how efficient it would be if it was operating at 20 degrees Celsius. And this one's not operating at 20 Celsius. It's operating at 17. So it's not going to have that much removal. Its removal is going to be brought down a little bit because the water's cold. So the way that we account for that is we would put in the efficiency at 20 Celsius, which here is 0.8914, and then multiply that by 1.035 to the power of 17 minus 20. So we're 3 degrees colder than what this formula is calibrated for. It's calibrated for 20 Celsius. So if it's colder, the bacteria are working more slowly. So that means that it's going to be removing 80% roughly of the BOD. So the concentration of the effluent from the first stage, you can see, is 31.4 milligrams per liter, because our incoming concentration was 160 and it's removing 80.4%. And so what remains is the difference between the inflow concentration and what was removed. So 160 milligrams per liter comes into the trickling filter. After a couple of rounds of recirculation, the stuff that's coming out of the bottom of stage 1 is 31.36 milligrams per liter. So then that becomes the inflow concentration for stage 2. What came out of the bottom of stage 1 is the concentration in of stage 2. So here's our formula for E2, and you can see it's the same basic equation as E1, but I've just specified that the concentration component should be the concentration that came out of stage 1, and then we also are going to be accounting for the efficiency in the first stage at the temperature that was specified. Okay, so stage 2 of the trickling filter is we're substituting in the same flow rate for the overall process. It has the volume. The second stage we assumed had the same volume, the same recirculation conditions. But you'll notice that the concentration that I'm putting into the second stage is the effluent concentration from the first stage. And then we're correcting for the removal efficiency in the first stage. So stage 2 would have a removal efficiency of 78% if it was operating at 20 degrees Celsius. But we need to, once again, correct for the colder temperatures. And so when we do that with this same formula of 1.035 to the power of the temperature, then you can see that that knocks the efficiency down to 70%. And so the final effluent concentration out of the secondary trickling filter is 9.18%. So these trickling filters are interesting. The equations that we use to size them are empirical. And their main advantage is just the vast reduction in electricity that they use compared to activated sludge. All right, that's all I have for you today. I'm going to stop the recording. I'll leave this up in case you're still writing down some of the numbers. But remember, meet in the lobby Wednesday a couple minutes before 1 for the tour of the treatment plant. I'll see you then.